How are chemotherapy drugs designed to protect healthy cells? Imagine fighting a fire with water that can sometimes also cool down the surrounding areas. That's similar to how chemotherapy drugs work to target cancer cells while trying to spare healthy ones. Chemotherapy drugs are mainly designed to attack cells that grow and divide rapidly, like cancer cells do. They interfere with crucial processes such as DNA replication and cell division, which are essential for cancer growth. But because some normal cells also divide quickly, like those in the lining of the bladder, strategies are used to protect these healthy cells during treatment. One key approach is to take advantage of differences in DNA repair abilities. Cancer cells often have faulty DNA repair systems, making them more vulnerable to damage caused by chemotherapy drugs like platinum compounds and topoisomerase inhibitors. Normal cells, on the other hand, usually have efficient repair mechanisms, such as homologous recombination and nucleotide excision repair. This means they can recover from some damage if given enough time between treatments, helping to reduce side effects. Another method involves temporarily stopping normal cells from dividing. Scientists have explored drugs that activate the tumor suppressor protein p53 in healthy cells. When p53 is activated, it causes these cells to pause in the G1 or G2 phases of the cell cycle, making them less sensitive to chemotherapy drugs that target dividing cells. Interestingly, some drugs can activate p53 without causing DNA damage, offering protection to normal tissues like bone marrow, while cancer cells with mutant p53 remains vulnerable. The body's own ability to detoxify also plays a role. Normal cells can metabolize chemotherapy drugs more effectively through enzymes like cytochrome P450 and glutathione transferases. These enzymes modify drugs into less harmful forms and help clear out toxic metabolites, reducing damage to healthy tissues. This metabolic difference helps protect normal cells during treatment. Timing and dosing are also crucial. Chemotherapy regimens are carefully planned to maximize cancer cell death while allowing normal cells time to recover. Treatment cycles are spaced out to minimize cumulative damage, and combinations of drugs are used to improve effectiveness and limit toxicity. Emerging research is exploring how harmful substances released from dying cancer cells can damage nearby healthy cells. For example, self-free chromatin particles can cause DNA damage and inflammation in normal tissues. Some agents, like resveratrol combined with copper, are being studied to deactivate these particles, potentially reducing systemic toxicity without adding significant side effects. In bladder cancer treatment, platinum-based drugs like cisplatin are common. They work by causing crosslinks in DNA, leading to cancer cell death. The difference in how cancer cells and normal cells handle DNA damage helps make these drugs effective and somewhat selective. Supportive care measures are also used to protect the bladder and other tissues from side effects during treatment. In summary, chemotherapy drugs are designed to protect healthy cells mainly by exploiting biological differences such as DNA, repair capacity, cell cycle control, and drug metabolism. Advances in understanding how these drugs cause toxicity are leading to new strategies that further shield normal tissues, making treatment safer and more tolerable for patients. Always remember, individual responses vary, so it's essential to work closely with healthcare professionals to find the best approach for each person.